So good morning, good afternoon, good evening, everyone, wherever you are. Welcome to the Omics Connect webinar series by PTR Biosciences. My name is Jenny. I'm currently the head of scientific marketing and training programs at PTR Bio. Today, I'm super excited to be the host of our session. And also, I'm very happy to have Hassaan Maan, who is currently the PhD candidate at the University of Toronto and Vector Institute, to be here with us and present about his recent study, which is very interesting, about the topic of single-cell integration. So his study characterized the impacts of dataset imbalance on the single-cell integration results and interpretation. Before we start, let me quickly go through the agenda for today to see what we have on the table. So we're going to spend the first few minutes to introduce to you about PCI Biosciences, who we are and why we're here hosting this webinar series. Later on, I'm going to pass the mic to Hassaan, who will be speaking about characterizing the impact of dataset imbalance on single-cell integration. And we'll save the last 15 minutes for the Q&A section. So if you have any questions along the way about PCI, about this Omics Connect webinar series, or about the presentation of Hassaan, feel free to leave your questions in the Q&A box, which is right below your screen. And we'll make sure that we'll go through them one by one in the Q&A section at the end of the webinar. All right. So if you are new here, welcome. We are PTR Biosciences, and we were founded as a company working on multi-omics analysis. So we were founded with a goal, that is to help you guys easily utilize the power of multi-omics, of combining multiple omics layers together. So we help you with that through delivering the software and delivering the service. So among our software and services, we currently have already introduced the CDM Multi-Omics Studio package, which is a web platform for integrating and analyzing single-cell, spatial, and other multi-omics data sets. And basically, you can import multiple data types into this platform, including bulk RNA-seq, single-cell RNA-seq, spatial transcriptomics, mutation data, proteomics, or metabolomics, all together into this platform, host them, and analyze each of the data set one at a time, like having some in-depth analytics into them. And also you can combine the insights from multiple studies together in order to gain bigger insights, like answering some validation questions, like finding common pathways, DE genes, or common targets that exist across omics experiments. And if you are interested in this platform, I included a QR code, which is at the bottom corner of the screen, so that you can quickly capture and request for a trial or demo of the platform. Meanwhile, aside from analyzing in-house data, it also supports you with analyzing public databases. So we do incorporate a lot of public databases in the platform for you to interactively access, as well as integrate with your in-house data. Those databases will include multiple spatial databases covering Visium, Cosmics, and other spatial technologies, single-cell RNA-seq databases from CellXgene, GTEx database for you to study expression of normal tissues, Recalc3 for analyzing bulk RNA-seq and single-cell RNA-seq data, and many more databases to come. Okay, and the reason why we started this webinar series is that we want to create a forum for everyone to get together and analyze and discuss about the topic of multi-omics data analysis, to hear about the latest developments in the field and also latest developments in our software and services. And if you are interested in PCI Biosciences and specifically this webinar series, you can also scan this QR code and follow us on our LinkedIn for upcoming updates. Okay, so without further ado, let's get started. I will pass the mic to Hassaan now.
Okay, thanks, Jenny. I'll share my screen. Okay, hopefully you can see that. It's all good.
Yeah, all good.
Okay, thanks, Jenny. And thanks to Pythia Biosciences as well as Jenny for the invite to present here. So essentially, yeah, I'm going to be presenting on our work that looks to characterize an aspect of single-cell datasets, which is dataset imbalance, and how that aspect affects the results of a single-cell integration analysis. So as an overview of the presentation, I'm going to start by just introducing single-cell sequencing and some of the associated computational analysis. I'll talk about why we do batch correction or single-cell data integration. And then from there, I'll provide the rationale for why it's important to study like dataset imbalance and imbalance samples when we're doing single-cell integration, and discuss the overall results of the study that we did. And then I'll provide some potential solutions to this challenge of imbalance integration and just, I guess, more of a discussion type thing, possible future steps in the single-cell integration field. So we know that if we have a given tissue sample and we take some sort of bulk molecular readout of that sample, for example, measuring bulk RNA inputs, we're going to obtain an averaged representation of the RNA expression across all the cells in the original tissue sample. However, by averaging, we're effectively masking out the individual cell heterogeneity or variance when looking at this from the bulk perspective. And so to determine that individual cell-to-cell variance or heterogeneity, we can employ single-cell sequencing and single-cell analysis. If we do single-cell sequencing at the RNA level, we'll obtain a distinct gene expression profile for each of the cells in our tissue. And this allows us to go beyond the bulk average level and identify things such as different cell populations and their variants and cell-to-cell communications, and do all sorts of interesting analyses that we couldn't in the bulk setting. So the following figure is just an overview of the overall computational analysis workflow for single-cell RNA sequencing. So after sequencing, you typically perform your upstream quantification and QC steps, such as base calling, demultiplexing, alignment, and then you end up with gene-level quantification and obtain a raw count matrix in the form of cell by gene, which outlines the gene expression for the different genes in each cell. Then we do some single-cell specific quality control, such as removing cells that have a higher amount of mitochondrial reads, because these are cells that might have burst during the process of isolation and sequencing, and so they end up with more mitochondrial than cytoplasmic reads. And after doing these steps, we obtain a process count matrix. It's between the steps of obtaining the process count matrix and actually clustering and annotating our different cell types in the data that data integration or batch correction is typically performed. Because this is done before annotation, typically we say that single-cell integration is an unsupervised sort of method or approach, meaning we don't have our actual labels for our cells at this point in our analysis. And then after annotating our cell types, we can do all sorts of interesting downstream analyses, such as trajectory inference or RNA velocity. So as we discussed, single-cell sequencing data is great in that we can use it to infer variance beyond the overall tissue level, but this does not come without its limitations. And one such limitation is that single-cell data is particularly susceptible to batch effects. Batch effects are effectively differences in measurement between samples and experiments that don't have any underlying biological cause. And they're due to technical factors such as different isolation protocols, different sequencing technologies, or even different sequencing runs or ambient conditions. These were first described in detail by Leek et al. In 2010, where this figure shows two batches of normal samples from healthy patients in the green and orange, And we can see that even after extensive filtering and normalization, at the end, the samples still cluster together based on the batch that they were processed in. And so we say that the main driver of variance in this data is the batch or technical effect, not any underlying biological signal. So given this, what do these batch effects actually look like in single cell data? This example illustrates just that. Let's say we take two batches or two samples of peripheral blood mononuclear cells, or PBMCs, from two different healthy patients, and we perform single cell RNA sequencing on them. This plot here shows the t-SNE, or the two-dimensional projection of all of the genes for each of the cells measured from each patient, here shown in the blue and orange. And we can see that the cells cluster very much apart based on the batch that they were processed in. We know that this should not be the case because these cells are PBMCs from healthy patients, so we know that they should share gene expression variants. And so we say that the main driver of heterogeneity or variance in this case is batch and not biological effects. We can employ some sort of batch correction or data integration technique to remove these effects. In this case, we've used mutual nearest neighbors. And after doing this, we can see that the cells from the different patients do, in fact, overlap quite a bit. And after employing this batch effect removal or integration, we can be more confident that the main driver of variance is truly biological and not driven by these batch effects. So I just discussed mutual nearest neighbors, but what is this algorithm actually doing? So just as an example, I'll walk through this. Let's say we have two batches of data, batch 1 and batch 2, that we know share some variance in the form of specific cell types. Here, the cells that are red and the cells that are blue. What mutual nearest neighbors will aim to do is to determine links or anchors between these two batches in the form of mutual nearest neighbors pairs. And this works as follows. Cell A from batch 1 is going to be a mutual nearest neighbor of cell B from batch 2, if and only if cell A falls within the k-nearest neighborhood of cell B, and cell B falls within the k-nearest neighborhood of cell A. So it's a cross dataset k-nearest neighbors lookup. After determining these pairs, we can effectively employ a subtraction process or a projection and project the data from batch 2 onto the data from batch 1. And then we can say that we've effectively undone this mapping that was caused by the batch or technical effect. And our data is ready for further downstream analyses after doing this. So I've used the terms batch correction and data integration interchangeably until now, but there is a distinction. And this distinction is highlighted in a review by Arjulaget et al. And in this review, the authors describe three modes of single-cell data integration. The first case is what's typically described as batch correction, which is horizontal integration. In this case, we have multiple samples which share the same features but are of different cells. An example of this could be single-cell RNA sequencing measurements of a similar tissue type but done on different cells. In this case, we're looking to use our features as our anchors and integrate across the cells of these different experiments. Vertical integration is the case where we've actually measured multiple modalities from the same cell. So an example of this could be simultaneously profiling RNA expression and chromatin accessibility from the same cell. In this case, our cells are in fact shared, and we're going to look to integrate across the different features in our data. Diagonal integration is the very challenging case where neither features or cells are shared. An example of this could be single-cell chromatin accessibility and single-cell RNA sequencing done on a similar tissue but of different cells. So given these different modes of integration, what are some limitations of current techniques? One ongoing limitation that's being further and further addressed by larger models is scalability. All of these assays, whether they're unimodal or multimodal, are continuing to grow in their throughput, and so methods to do integration have to scale appropriately. The next is interpretability. Often those techniques that are very scalable and expressive have uninterpretable integration results. An example of this could be methods that utilize deep neural networks, and they'll be quite hard to actually interpret how they're doing the integration. And the last limitation is the one I'll focus on for the rest of the presentation, and the one that we focused on for the work that we did, which is robustness to dataset imbalance. So imbalance here refers to a different number of cells, different cell types, or different cell type proportions between the samples or batches that we're looking to integrate. Previous benchmarking techniques have shown that methods very much struggle in the cases where the samples that they're integrating are in fact imbalanced, and they tend to lose a lot of biological information. Dataset imbalance is relevant in many fields, and I'll talk a little bit about this a bit further. But you can think of examples such as development and tumor biology. In development, we have differentiating populations and also depletion of stem-like populations. And so if you think of sampling multiple developmental time points, you are not likely to share a high degree of cell type variance. And in tumor biology, of course, we can have both intra and inter-tumor across different patients variance. And that could be low or very high depending on the tumor type that you're looking at. Because, as I said, we do the integration step typically in an unsupervised manner, it's hard to know or define the imbalance before doing our annotation. So we typically don't have this information when we're doing the integration. So the focus of the work that we did was really on this last challenge, and we focused it on the specific case of horizontal single-cell RNA sequencing integration. So yeah, just some preliminaries before I get into the actual results. If you're an end user of integration techniques, you might be wondering, okay, well, how do you know if your data set is imbalanced? Once again, because this step is typically done in an unsupervised manner, usually don't know. Of course, having some biological prior knowledge will in fact help. For example, if you're looking at tumor data for multiple patients and you know a priori that this type of tumor has a high degree of interpatient variance, you can maybe take that into account when before doing your integration. And I'll talk about that in some of the guidelines that we have for this sort of setup as well. But a challenge with that is technical effects can also mimic imbalance. For example, if one technology doesn't capture one cell type really well, right, that cell type might end up being underrepresented or misannotated. So that sort of challenge also makes it even more difficult to know this imbalance a priori. Then the other question you might be wondering is, are these multi-sample, multi-batch data sets typically imbalanced? And we would argue for a large chunk of the important biological questions they are. So I already discussed tumor data because you can have patient as well as patient-to-patient variation, as well as variation within just one tumor itself, depending on where you're taking the biopsy. Developmental data I've also discussed, and this figure here actually shows an example of imbalance between measurements and developmental data. This is a data set of hindbrain development from a study from Vladou et al., which are some of our collaborators. And effectively, this t-SNE here shows the different annotated cell types across the different samples for this experiment. The next slide shows the actual different samples. So samples were taken from mice that were sacrificed at different embryonic and postnatal time points. And you can see if you flip back and forth between these two plots, some cell type populations actually only exist in one time point, which is a highly imbalanced case if you're considering all of this data together. So that just illustrates one example of where you might find imbalance in actual real biological data. And this is pretty much prevalent through most of development, we would say. So I talked about benchmarking studies as well. So previous benchmarking papers have suggested that integration techniques do in fact struggle in imbalanced data sets. Perhaps the most striking example I could find was from this major benchmarking paper from Tran et al. That was published in 2020. And this result is across the different methods for integration. And it looks at, on the y-axis, how well do the methods mix the batches? And then on the x-axis, how well do the methods essentially preserve biological information? And you have to keep in mind that this is a very highly imbalanced data set that's being integrated. we can see that the scores on the y and x axis range from 0 to 1, 1 being the best. Most methods mix the batches quite well. However, in this imbalance case, very crude way to look at this is that most methods are losing up to 70% of the actual biological information when doing this integration. So that's, sort of an example that shows you how integration can go wrong in these imbalance settings. So for our work, what we wanted to try to do is to better characterize what we found here. The first thing is to actually measure the downstream effects of imbalance. So not just looking at the integrated space, but actually looking at, does this information loss affect biological conclusions you might make after integration via the various downstream steps, such as trajectory inference, differential expression, and so on and so forth. We also wanted to use better metrics than the ones used in this study and other studies, and to try to look at things at a cell type specific manner. So to look at which cell types is the information being lost in, because that's also something that users of these methods would be interested in. Also to create a controlled way to test the effects of imbalance, and I'll talk about this shortly. And then lastly, see what solutions we can potentially come up with for the problem of imbalance integration. So the following slide, depending on whether or not I might lose you in subsequent slides, is just like a summary of the information and the main takeaways from the work that we did. So this work was done jointly with members of the Bo Wang and Karen Campbell labs, and I'm very grateful for the help of Lynn, Chengsing, and Michael especially, and of course this Bo and Karen supervision. So as an overview, we find that dataset imbalance leads to significant variation in all of the downstream analysis that we tested after integration. What this basically means is if you had an imbalanced case for the same data versus imbalanced samples, you are going to have a different result in your integration space. You're going to lose information in the imbalance setting. We also found that this can actually, in fact, lead to changes in the biological interpretation of your results. So how you interpret your downstream analyses. And we showed this in an example using pancreatic ductal adenocarcinoma data for tumor normal calling. Unfortunately, I can't get into that analysis for the presentation, but if you're interested, all that work is in the paper. We found that it's not just the imbalance itself, but two sort of factors that lead to these quantitation differences. One is, in fact, the imbalance, meaning cell types that are less prevalent are more likely to have information lost about them. But the other is about how closely cell types are related to each other in the modality that you're measuring. So for example, if you're looking at RNA space, how transcriptomically similar are the cell types? And those that are more transcriptomically similar are more likely to have their information lost in these imbalanced integration settings. As one set of solutions for benchmarking, we introduced these novel clustering metrics, which weigh rare cell types equally as prevalent cell types. So we don't want the scores for integration to be dominated by the most prevalent cell type. And we also provide a series of guidelines for end users of integration techniques in these imbalance scenarios. So the following figure goes over an overview of the work that we did, sort of from figure 1. So what we did is we took 2 control and 4 real-world complex single-cell RNA-seq datasets. Control in this setting means we took actual data and we subsampled it such that the batches that we're integrating are completely balanced, meaning that the cell types and cell type proportions are equivalent. Then we integrated that data, and we also did a series of integration perturbation experiments. And perturbations in this case are not the classical single-cell perturbations you're thinking of, but they're actually artificial changes to dataset imbalance. So we induce an artificial imbalance by subsampling or downsampling specific cell types. And then we compare those results after integrating with the control measurements. So in terms of quantifying the results, we do the typical thing that previous benchmarks have done, which is how well does the integrated space preserve biological signal and how well does the integrated space do batch correction. And then we further quantified the impact of these changes on the downstream analyses, including clustering, differential expression, trajectory inference, k-nearest neighborhood classification, and curator reference projection. For the sake of time for this presentation, I'll only have time to get into the k-nearest neighbors classification and marker gene stability experiments. So to begin, the k-nearest neighborhood analysis was really our experiment 0. So once again, we sought this controlled environment for testing the effects of dataset imbalance. So what we did is we took 2 PBMC datasets, so 2 batches from, once again, 2 healthy patients. These were processed using 2 different technologies. And we subsampled the data such that the cell types and their proportions are completely equivalent between the 2 batches. We can see that based on the UMAP projection of the data, there is in fact a batch effect between these 2 samples. Then what we did is we just integrated this data as is, as our control, and we did the perturbation experiments. So for the perturbation experiments, what we do is we randomly select one of the 2 batches, then we randomly select one cell type in that batch, and we either downsample it to 10% or completely remove it from that batch. This does not mean that the cell type is removed from the experiment, because remember, it's still present in full proportion in the other batch. And then we perform integration on this perturbed scenario. And we're looking to compare the results of the perturbed scenario to our control setup. So once again, as our experiment 0, we really looked at the integrated space and how well does it preserve information at a cell type specific level. And so to do this, what we did is using our control experiments and our perturbed scenarios in the integrated latent space, we trained a K-NN classifier on 70% of the data and held out 30% of the data for testing, for annotating the other cell types. What this plot is showing is the K-NN classification F1 scores based on the cell type that was affected, so which cell type was downsampled or ablated, and comparing that to the control for that subset. So this figure here shows for each different method, if we downsample or ablate B cells, how is their K-NN classification score actually affected? For B cells, we can see that for the most part, it's not affected. However, there are marked decreases in performance for CD4 and CD8 T cell subsets, as well as the 2 monocyte subsets, and to a lesser extent, the NK cells, where K-NN classification performance does decrease after we do this downsampling and induction of an imbalance. So we're losing information in that setup. However, we were curious as to why the B cells performance was not affected. And so we hypothesized that this might be due to the transcriptomic similarity of the cell types, because there's 2 T cell subsets are very transcriptomically similar, as are the 2 monocyte subsets, and NK cells are very transcriptomically similar to the 2 T cells. So we did a follow-up experiment where all we did was we hierarchically clustered the cell types into B cell, monocyte, and NK/T cell subsets. And we repeated all of the perturbation experiments as well as the control. And we found that we actually are not losing information at this higher level of hierarchy, because at this point, the cell types are very transcriptomically distinct. So the takeaway here is that it's the sort of synergy between transcriptomic similarity and dataset imbalance that's leading to these changes in interpretation of the results post-integration. So the other analysis that I quickly just want to show is on marker gene expression. So this plot is pretty loaded, but I'll quickly walk through it at a high level. So what we did is we once again did the control experiments for the PBMC data. And instead of just looking at K-NN classification now, what we did is a differential expression analysis. And we want to compare if the ranking of the marker genes specific to certain cell types changes before and after these downsampling and ablation experiments. The reason why we consider the ranking of marker genes is because the ranking of markers is typically used to actually annotate our clusters. We do a 1 versus all differential expression analysis between our clusters typically, and whichever marker genes show up highest in the differential expression are used to rank the different clusters essentially. So what these plots show is that if we just look at SCVI's results, for instance, if we downsample B cells or CD4 T cells, the ranking of the marker genes specific to those cell types do in fact change quite significantly in a post-integration differential expression analysis. We found this to be statistically significant, as well as all of the other downstream analyses that we tested. And a supplementary analysis, which, and again, unfortunately don't have time to show, we did actually see that this can lead to changes in actual annotation and results interpretation if we're using a marker gene ranking list to do the annotation. We found similar results in all of the other downstream analyses that we tested. Once again, unfortunately, I have to admit it for time purposes. And we also went through an extensive biologically motivated example using tumor data and trying to annotate normal and tumor epithelial cells and showing how imbalance can in fact change the interpretation and conclusions in that sort of setup. So if you're interested in that, definitely encourage you to check out the rest of the results in the paper. Okay, so I talked about potential solutions. The first solution that we really want to introduce was from a benchmarking perspective. The challenge is that the current metrics that are used to benchmark integration methods really don't consider the proportional differences between cell types. And what that basically means is that cell types that are highly prevalent will dominate these scores. If you've done any sort of like statistical modeling or machine learning exercise, this would be akin to using accuracy instead of a balanced accuracy. The challenge is that the balanced metrics didn't exist, at least before the work that we did for benchmarking. And so just to walk you through what's typically done in a benchmarking scenario for integration is, let's say you integrate your data with some method. What you typically do is you do unsupervised clustering after the integration step. This is done to simulate the actual annotation step in a single cell analysis. And then you're going to look towards the actual labels that you have, because we're just pretending we don't have the cell type labels in our experiment. And we want each cluster in our integrated space to only correspond to one cell type. For the batch labels, we want the opposite. We want each cluster to mix the batches together a lot, because then we would assume that any technical or batch factors are removed. That's why we take 1 minus the batch label scores typically for any metric. So an example of the metrics that are used is the adjusted RAND index. This is used quite frequently. What the adjusted RAND index is measuring is, okay, if you do your unsupervised clustering after integration and you end up with some set of labels, how well do these labels map to your actual cell type labels? So you want there to be a one-to-one mapping between your cluster labels and cell type labels. If there's any discordance here, the adjusted RAND index score is going to penalize this significantly. But again, the challenge is that this index does not have any idea of the proportions of the different cell types, right? If one cell type is dominating this list, that would essentially mean that it's going to dominate the RAND index score. And so what we did is we tried to come up with balanced versions of these metrics, and which includes the adjusted RAND index, adjusted mutual information, and a few others that are typically used. So an example to show how the balanced versus base or imbalanced metrics work is shown here. Let's say we have 3 classes of data, class A, B, and C, where class B is present in a much smaller proportion compared to the other 2 classes. You can think of this as a rare cell type in a single cell scenario. And let's say we have, we do an unsupervised clustering step, and we've misannotated class B, meaning we've mixed it together with class A. If we look at the results of these metrics for the base imbalanced metrics, we call them imbalanced, but they're just the metrics as they were, we can see that the scores are not really penalized that much overall. But if we use the balanced version of the metrics that we came up with, we can see that because they weigh the proportions of each class or cell type equally in this analysis, this will penalize as a result significantly, because we've essentially misclustered or misannotated class B. And so we believe that these metrics should be used in future benchmarking scenarios, because often in biology, we don't just care about the very prevalent cell type. in many cases, we might actually very much care about more rare cell types. The other set of solutions that we proposed was really for end users of integration techniques. So the overall impacts that we observed were not method specific. So we weren't going to try to, develop a method-based approach to these guidelines. Instead, what we developed was at a high level, an iterative process where before integration, end users can try to gauge whether or not there might be imbalance present in their data set through a series of heuristics. And depending on if these heuristics do indicate there is imbalance present, the users can tune the integration techniques at the integration step. And we have various things you can do with different integration techniques and supplementary material, as well as the example in the paper, where if you have an imbalance, you can try to tune the methods to better preserve biological information. In the post-integration stage, what we're really looking at is the trade-off between batch mixing and preserving biological signal adequate for our analysis? And we also provide some heuristics on how you can actually determine how much information you might be losing in this setup. If it is, the user can continue towards downstream analysis. If it's not, the user can return to the integration stage and try to tune the methods further. This figure is a bit abstract and high level, of course, but this is accompanied by sort of broader description and supplementary materials. But more importantly, we have a full code-based tutorial for these guidelines that is present in the repository. And yeah, we always are happy to hear any feedback that the community might have towards these current guidelines, anything else that users think we should add, any sort of other pain points when it comes to this sort of a scenario. So if you're interested, feel free to shoot me an email or open up a GitHub issue or anything like that. Okay, so given all of this, what does this actually mean for single cell integration overall? The key point that we're trying to emphasize here, once again, is that in these imbalance scenarios, we are losing biological information. And so it's really important to try to factor this in when you're thinking about doing integration for your data. And also factor in this cell type similarity property that we found. We also think that this is really important. Like, let's say, for example, you're trying to determine very, very fine-grained T cell states, and you have many, many different samples which might not share variants. That might be a scenario where you might want to pause and say, okay, do I want to integrate? How do I want to integrate so I don't lose information? Because I'm trying to find these very, very fine-grained cell states between and within my different samples. Because once again, this result is method agnostic. We don't believe that users should just rely on a cookie cutter method, like, okay, let's just use SURAT, let's just use SCBI. I really think a deeper investigation has to be done, which hopefully our guidelines can help with, but also internally within your research team on how to go about this. And once again, going forward, we believe addressing these effects is important. So that's why we came up with the guidelines. And we also think better benchmarking in the future is going to be important. As far as we're aware, we're the first study to sort of look at the downstream analyses post-integration and to study the effects on integration on those downstream analyses. We really think in terms of future benchmarking studies, regardless of topic in the single cell or just omics field in general, it's really important to actually look at the results of the downstream applications that you have after you obtain your representation, whatever, from whichever analyses or methodology that might be. So in terms of the future of integration, we know that foundation models have become very popular in the field. We're currently looking to test these foundation models in some of the scenarios that I described. We don't have any strong prior on whether or not they're actually going to work or not. The only reason why we think that they're worth testing is that a pre-trained foundation model is quite a bit different from a purely unsupervised approach, right? most of the like all of the methods that were used in this paper are unsupervised integration techniques, like Seurat, like SCVI, like Harmony. A pre-trained foundation model has some understanding of context. And so we think that that context might actually be able to help in these scenarios, but we're still testing that internally. And of course, with these foundation models, there is currently a lack of rigorous benchmarking studies like that have been done for single cell integration previously. So we're also looking to see that more in the field and to actually see whether or not they are in fact, able to obtain novel insights beyond just like a increase in score for these different downstream tasks. Once again, really want to hamper on the points of including downstream tasks in benchmarking. If you're interested, a really, really great example of this is the glue benchmark in natural language processing. I think that's one of the gold standards for having downstream tasks and actual conclusions of your analysis, and basing how good your representation is based on those downstream tasks. We also believe that future methods should not just simply look to benchmark and do better than other techniques, but to also answer questions of like applicability, such as when should end users actually perform integration, right? When should they be wary of integrating or doing any sort of other single cell analysis? I think, at least from my perspective, there's a lot of work, really great work that's often overlooked in the field when it comes to applicability. An example of this is the paper by Lee et al., where they look at this sort of trade-off between conservation of biological signal and batch effect removal in deep generative models like SCVI, for instance. We really think that this type of work is quite important. And because end users don't just want to know like, okay, which method is doing best on which data sets that are being used for benchmarking. They want to know how should they approach this task overall with respect to their data, with respect to the prior knowledge that they have about their data, and what is going to go into and be potentially lost from the representation that they're obtaining through these sorts of analyses. So yeah, I just like to thank my supervisors, members of the Bo Wang and Kieran Campbell labs, especially Lynn, Michael, and Shengson, as well as the various consortia where we used data for this study from. And yeah, if you're interested in checking out the paper, the link is there. I guess I don't know if slides are going to be disseminated, but in search, it's pretty easy to find. And if you're interested in the balanced clustering metrics, that GitHub is also in the code and data availability for the paper, as well as the guidelines and overall pipeline for reproducing these results. And that's it for me. Thank you for listening.
Okay, so thanks Hassaan for your wonderful presentation. I think it's time for the Q&A session. So I will read out loud some of the questions we have currently in the Q&A box. Again, if you have any questions, feel free to leave them in the Q&A box below the screen so that I can read out loud for Hassaan, or you can raise your hand and we will unmute you so that you can directly chat with Hassaan. So I think the first question is there any statistics to really confirm the imbalance of data? How much difference is considered imbalance?
Yeah, that's a great point. Yeah, I guess what you're sort of asking is like, I went over this initial definition, right, where we're considering like, okay, different number of cells, different cell types, different cell type proportions. But if you're asking whether or not there's like a threshold for saying like, okay, my data set is balanced versus my data set is imbalanced, I wouldn't really say that this is the case. I would say based on the results of our analysis, which were, if you really look at it, like if we go back to like experiment 0, like here, right? All we're doing to induce an imbalance is for one cell type, we're downsampling it to 10% or removing it from one batch. I would say that in itself can be thought of as like a good heuristic. Like, okay, if one cell type is present in much lesser proportion are not present in one batch that you're integrating. But in terms of like a hard definition, like, okay, this is balanced and this is imbalanced, I would say there isn't. You're probably looking for an answer like, okay, at which point do I have to be concerned about losing information? I would say if you have both factors, like both, okay, you have an imbalance, like one cell type is present in a, let's say, maybe at least 50% lesser proportion to other cell types, and you have that similarity, that's when you can begin to start thinking, okay, it might be problematic. But just for the strict definition purposes of imbalance and how that relates to information loss, we haven't really come up with like that strong of like, okay, a threshold, like, okay, this factor, this level of imbalance is going to lead to biological information loss. Because once again, it is the combination of two different properties. So it becomes even more difficult to sort of define like a very clear threshold. Hopefully that answered the question.
Yeah. Thanks, Hassaan. Another question from Sirisha. With the foundation models, what is the minimal representation of rare cell types required in your experience?
Yeah, so it's a great question. Unfortunately, the answer right now is we don't know. We're just testing these sort of things right now when it comes to the foundation models. I think there was some work done in SCGPT, like for integration, of course. But from what I know, and I should know because I was put as an author on the paper, there wasn't actually rare cell types investigated. For the other foundation models, I'm not so sure. Like I know there's also like gene former, and now there's like niche former for spatial data. So for that, I actually don't know if rare cell types were investigated. So yeah, sorry. Unfortunate short answer is we currently don't know because we haven't done this analysis for like the rare cell types, at least as far as as far as I'm aware, I don't think it's been done in the literature. But if you know of like a paper that's looked at that, I would be very interested in checking it out.
Okay. Thanks, Hassaan. Another question is that, do you think we should control for cell type annotation step? Since I think it's crucial for our integration decision.
Control for cell type. Yeah. Sorry, I guess I don't exactly know what you mean by control for cell type annotation step. If you're like, if you're here, maybe you want to like jump on the mic and like clarify.
Yeah, so if you want to directly ask Hassaan and clarify that question, please raise your hand so that I will unmute you. So Hassaan, I think what the person means over here is that should we, quality control the cell type annotation first before we decide or continue with other fine-tuning step.
Right. Okay. Yeah, I think so. I think from my perspective, let's say you do annotation in a few samples of your data. Let's say you have like 10 samples and you do annotation in like 2 or 3 samples. Let's say you find an imbalance when you do that individual sample annotation. From that perspective, yes. I think that means like you probably should be potentially wary of doing integration, or you should try to do an integration that tries to better preserve biological information. I think, yes, that can definitely be a step that can be taken. And in fact, in the guidelines, we sort of, if I can just go to them very quickly, we sort of do have a heuristic, like, okay, if you have a reference to do annotation of individual batches, you can do that. And if you find that there's an imbalance, you would probably want to maybe think about, have second thoughts about doing integration, or if you're doing integration, tune the methods to better preserve biological information. So yes, from that perspective, yes, I completely agree. I think doing that annotation step in individual batches a priori would be a good approach. Unfortunately, I think a lot of research centers and research labs may not have like the resources to do that, especially if you have like a lot of data and whatnot. So it can become tricky, of course. But yes, I think if you have the ability to do that, I think it can be a good first step for sure before integration.
Yes, thanks, Hassan. Well, I think one more question actually that is for me. Is there any guideline to differentiate whether the downstream analysis results derive from the biological differences itself or the dataset imbalance?
Right, from the biological differences. Yeah, so I think that's a great question, right? And I think unfortunately, let's say you do your entire integration pipeline, you don't like try to assess whether or not you're losing information or potentially have a data set imbalance anywhere in your setup. If you don't do any of that, you won't be able to tell, right? Because you can have some sort of biological differences present in your analysis, but then if you don't try to gauge whether or not those biological differences are essentially being driven by loss of information or their actual real biological differences, it's really, really hard to actually tell. And so I think in terms of the guidelines that we have, really what they try to offer is there's no way of exactly knowing, right? Especially before you've annotated your data. But we hope that if the user sees that there is imbalance present, and they also sees if there is information being lost potentially post-integration. So we have a heuristic actually for assessing whether or not you're losing information post-integration. And that's in the guidelines as well as like the code-based tutorial. So in one sense, you can use that to try to infer whether or not your downstream analyses results are being driven by biological differences or information loss due to data set imbalance. It's always going to be hard to tell exactly because, like I said, defining imbalance a priori is in fact challenging, and the problem is there's always this sort of coupling, right? We go back to this step, right? There's always this coupling of the data integration step and annotation, right? So they're sort of dependent on each other, and so are the downstream analyses, right? So you can't really uncouple the integration with the downstream analyses. So that's just to say it's hard to know exactly whether or not it might be due to the imbalance or actual biological differences, but we're hoping that the guidelines can offer at least some information related to that. I think, yeah, just to like end off on that point, I think integration, this has probably been mentioned in a lot of papers, integration is always the trade-off between removing technical effects, but also removing biological information. That is always going to happen. And I have like an interesting like sort of perspective on this with like decision theory, right? Like if you ever look at decision theory, what decision theory basically means is that you're trying to essentially minimize your essentially, you can look at it from a minimization or maximization perspective. But what you're saying is like given some set of actions, you're trying to basically take the maximum or minimum expected value based on what you're looking at. And the expected value in this case might be actually preserving biological signal, right? Because batch effect removal is also trying to preserve biological signal because you don't want to introduce technical effect-based biological signal, right? But then with integration and imbalance integration, you're trying to prevent loss of biological information by not overcorrecting, essentially, right? So in a sense, it's always up to the end user, and it's up to their model of the world based on the information that they have available. So I think that's where prior knowledge of your data can really help. And if you don't have great prior knowledge, that's where the heuristics and the guidelines and other heuristics that users might use can really help. Yeah, sorry for the very long-winded answer.
Yeah, thanks, Hassaan. So another question, have you extrapolated any of this work to identify cell states beyond cell types? Thanks, this is very useful work.
Yeah. So that's a great question. So I think in a sense, like for me, the sort of boundary between cell type and cell state is not very clear always, right? Because some people might think of like, okay, a CD4 positive is a cell state of T cells, right? It's like a subclass of T cells. I think one thing we can say for certain based off of like this property that we've discovered, right? Like, this overall transcriptomic similarity will lead to loss of information. We haven't gone very, very deep into like very fine-grained cell states. And one of the reasons is that we already found like a loss of information at these sort of higher states like CD4 versus CD8 T cells, CD16 versus FC3R. I always forget what subset, but the two monocyte subsets, right? We haven't done this, but what we hypothesize is that as you go further and further into these more fine-grained cell states, you're more and more likely to lose information about them, essentially, if you have that imbalance present. So yeah, while we didn't do it, that's what I sort of think would end up happening if you went into like very fine-grained cell states.
Okay, thanks, Hassaan. So the final question of today is, do you plan to make the package to test the imbalances between batches?
We tried. It's a tricky problem, right? We tried, and I think we're still trying. Um, the essential challenge that comes with this is that if you don't have any information like a priori on like, because you're doing this in a purely unsupervised manner, right? So you have to think of this challenge as like, okay, I have two data sets. I don't have any annotations for them. I want to essentially predict if there's an imbalance. Well, in a way, that's sort of like actually doing the annotation step, if you think about it, right? Because you can't predict an imbalance without having like at least some degree of annotation. So when we tried doing this, we sort of got caught in this loop of like, okay, do we do like some sort of reference-based annotation? But okay, if there's an imbalance, we've shown that reference mapping might not work that well, and so on and so forth. So I think I would say it's ongoing work, but it's particularly challenging because not having any prior knowledge makes it quite limited. But it is something that we're continuing to try to investigate, and to potentially being able to do automatically in the future. Yeah.
Okay, thanks, Hassaan. I think that's all the questions for today. Yeah, thanks again, Hassaan, for your very nice presentation and an engaging Q&A session. I myself enjoyed it a lot, and I hope the audience today enjoy it as well, as much as I enjoyed it. Okay, so thanks again, and have a nice day, everyone. Thank you, and bye-bye.
Sure. Thanks, Jenny. Thanks, everyone. Bye.
Thanks, Hassaan. Bye-bye.